we are going to discuss the last part, that is the mechanism of replication part. In previous two parts already I am discussing about the introduction of replication, DNA replication, and their origin point, and the next part discussing about the proteins or enzymes involved in this replication process. Today in the last part we are going to discuss the mechanism of DNA replication, how this DNA replication occurs. Already we discussed regarding the origin point, the point from where replication starts. Okay, in case of prokaryotes, that origin of replication, it is generally from a single origin point where replication start. In case of suppose bacteria, in case of bacteria there is a circular genome is present, and that circular genome is replicated from a single origin point. But in case of prokaryotes, there are multiple origin points are present. Okay, in case of eukaryotes, there are multiple origin points are there. Okay, and that origin point may be bidirectional or may be unidirectional. So what is bidirectional or what is unidirectional? Suppose there is a DNA strand, and here suppose this open up. Suppose this is the origin point, origin of replication. If replication starts only in one directional, then that is called as unidirectional. Or suppose a double stranded, let's just open up. Suppose here origin point is here in that point, and they start this direction also to this direction also, then this is called bidirectional DNA replication. So after that we'll discuss the different steps involved in DNA replication. Here some major steps are initiation, elongation, last one is termination. Initiation phase already we discussed that one. There means at origin point that this proteins or enzymes should be bind and that's open up. Okay, and that is the initiate process. Okay, that is called initiation process. And another some steps here we are discussing. Also here we are discussing the functions of another enzymes like primase, okay, phosphorylase here. Okay, so initiation is adapted, identified at origin point. Now another some steps we'll discuss here that is activation of deoxyribonucleotides. These are very important steps because in DNA replication the nucleotides have to bind with the help of this phosphodiester bonds based on the strand, single strand. Okay, that means suppose there is a double helical structure. Suppose here adenine, thymine, guanine, cytosine, cytosine, guanine, thymine, adenine, like that, or guanine, cytosine. So when we break down with the help of this helicase enzyme, suppose here adenine, thymine, guanine side. Suppose here this thymine, adenine, guanine, cytosine. Now here suppose this DNA synthesis occurs. So suppose here guanine is there, then it will join cytosine. Thymine is there, then it will suppose join adenine. Suppose cytosine, then it will join guanine. So between this adenine and cytosine, what bond is present? That is called as phosphodiester bond. And between this and this strand, these two are joined by with the help of hydrogen bonds. Like here also, here cytosine is there, so it will join guanine, but here adenine, so thymine. So between thymine and guanine also this bonding required, that is bind with the help of this phosphodiester bond. Okay, that means now we are going to discuss what is actually happen in activation step. This deoxyribonucleotides means we have 4 types of nucleotides. These are called as deoxyadenosine monophosphate, deoxythymine monophosphate, deoxyguanosine monophosphate, okay, deoxycytidine monophosphate, that means DAMP, DTMP, DZMP, DCMP, that means adenine, thymine, guanine and cytosine. These are the nucleotides. But in nucleoplasm, these are present as a monophosphate. Okay, to take part in replication process or to activate another 2-phosphate is required. Another 2-phosphate group is required. And that steps is done by this transfer of phosphate group or addition of phosphate group. One enzyme is there that is called as phosphorylase. Okay, that enzyme is called as phosphorylase. Phosphorylase is the enzyme which aid the phosphate group. That means to become activate. Because when they synthesize, they synthesize as deoxyribonucleoside monophosphate. Deoxyribonucleoside monophosphate. Here I am written deoxyribonucleotides. Here I am writing deoxyribonucleoside monophosphate. Here I write side, here tide. Why? Because here separately I write about the phosphate. How many number of phosphate? Single phosphate. That's why it is monophosphate. Don't write deoxyribonucleotide monophosphate. That will be wrong. Because when nucleoside containing phosphate, then as a whole it is called as nucleotide. Without phosphate it is called as nucleoside. That as here I am writing separately, that's why here written deoxyribonucleoside monophosphate. How many number of phosphate? Single phosphate. So to become activate, you have to convert it into which form? Triphosphate. That means having 3 phosphate groups. Okay. So how it occurs? So here this mono to tri, that means addition of phosphates, another 2 phosphates. So that is done with the help of this enzyme phosphorylase. So how they bind? In case of nucleotides, you know that one sugar is there, another one nitrogenous bases are joined, that may be adenine, that may be thymine, guanine, or maybe cytosine. And this is joined with the help of this glycosidic bond. Okay, and here this is the pentose sugar or called as deoxyribose sugar in case of DNA. Why? Because oxy group is missing in carbon position 2. What is carbon position 2? Here this is carbon 1, that is carbon 2, that is carbon 3, and that is carbon 4. Here that one is the carbon 5. Okay, now with this 5 carbon, these phosphates are joined with the help of ester bond. Suppose sorry, here suppose I am giving 5 here, so CH2, suppose OH group is there. So another phosphate, you see phosphate, this is OH group, then this one. Now OH and OH, that will combine and release water molecule. And now we can tell this one, suppose here I am showing the OH group because that is, we are talking about DNA not RNA, because in case of DNA this oxy group is missing, that's why it is called deoxy. So here you see, so this water is released and this phosphate is joined with 5 carbon position. Now it is called as monophosphate. If another 2 phosphate is joined, suppose another phosphate, this is again this OH, O minus, O, OH, okay, same way again this releases water molecule, this is become diphosphate, and again OH, again the water molecule is released, so it become triphosphate. It become triphosphate. Previously it is monophosphate and another 2 phosphates are added. Okay, so this is the function of this enzyme phosphorylase, addition of this phosphate group. Now it is become activated. Now it is converted to from, suppose here, suppose adenine is present. Now it is converted to from deoxyadenosine monophosphate to convert to where? Deoxyadenosine triphosphate. Suppose here thymine is present. So it is converted to deoxy TM, instead of M, here we write P, that means TTP. This triphosphate like that, GMP, GTP, CTP, it is also converted to deoxy GTP. This is converted to DCTP. That means all are converting to in triphosphate form. How? In that way. They are joining the phosphate. So here that's the function of this phosphorylase enzyme. Okay. Now another enzyme, right, that is primase, where I left in the part 2, that will be discussed in mechanism chapter. Now I am going to discuss the function of this enzyme primase. Already I told that primase is the enzyme which synthesizes primer. What primer? RNA primer. What is primer? Some short nucleotide sequence. Okay. Now, why need these short nucleotide sequences? Why need? Because what is the function of DNA polymerase? It will add nucleotides one after another. How it will add? Based on the complementary strand, because the mode of replication is semi-conservative. So because suppose here thymine is there, so it will add adenine. Suppose guanine is there, it add cytosine. To like that, another strand will produce. Okay, according to complementary. But suppose here adenine is there, it will add thymine. Suppose guanine is there, it will add cytosine. So between this thymine and cytosine also this phosphodiester bond is required. Okay, and for that, this OH group, at carbon position 3, that OH group is required. That means, suppose I am showing, suppose here, suppose nitrogenous base adenine is present, one nucleotide. Suppose next nucleotide is suppose guanine. It have to come and bind with this carbon position 3, because DNA synthesis occurs in which direction? 5' to 3' direction. Because the enzyme DNA polymerase have the capacity to synthesize DNA in 5 to 3' direction only. Okay, so here this is the OH group. So another one nucleotide, suppose another one, suppose I am showing here, suppose guanine, nitrogenous base, suppose guanine. It containing the same way, containing this carbon. Here also these phosphate groups are there, joined. Okay, like that. These phosphate groups are there. Another 2 phosphate is there also. Another 2 phosphate. Suppose this is another 2 phosphates. I am showing it separately. Or now, with the help of this ester bond, these are joined, this oxygen is present in between these also releases water, this is water. That means there is another nucleotides. Suppose it is number 1, this number 2. Now let's come to join with this carbon position 3. Now it is the carbon position 5. Okay, and here also another OH group is there. Now it will come, and what will happen here? It will release this phosphate. Just I am here labeling. This phosphate is called alpha. This is called beta, and this is called gamma. Here also I am labeling. This one is alpha, beta, and that one is gamma. So what will happen? These two phosphates will be released. Okay, these are the pyrophosphates, 2 phosphate that will be released, and only this phosphate will bind with this. Okay, with this, which this 3 carbon position. That means these 2 phosphates are released. Okay. Now this phosphate the release is also produce water, and it will be released. And with this, now with this I am showing here a little bit here. So with this is the phosphate. So with this of this, it combined with this carbon position 3. That means that is called as phosphodiester bond, because with the help of this phosphate, this combined with these nucleotides. With the help of this phosphate, it is combined with another nucleotides. So for addition of these nucleotides, DNA polymerase required this OH group. DNA polymerase required this OH group, hydroxyl group. Okay, if suppose this is double-stranded DNA, so it is open up. Now in that way, directly, suppose here adenine is there, suppose thymine is there, guanine is there, directly DNA polymerase unable to give thymine here. So it at least it required a short double-stranded region. Okay, why? Because DNA polymerase cannot, suppose here thymine, so it cannot directly add here adenine. Okay, because DNA polymerase needs the free OH group. So with the help of this primase enzyme, they synthesize short nucleotides, which according to complementary, here this primers are bind. Later on that will be removed. That will be discussed in the elongation part. Later on that will be removed. Okay, so when these short nucleotides are bind, that will provide the OH group, free OH group like that here. So according, suppose this guanine is present, now DNA polymerase is able to add cytosine here and join with, suppose adenine is present, between cytosine and adenine this phosphodiester bond is formed, and in that way the process continues. That means why primer required? To give the free OH group. Otherwise DNA polymerase unable to add nucleotides taking a single strand DNA. Okay, that means based on that single strand DNA, directly it cannot add nucleotides according to the complementary. It needs the OH group, and that is provided with the help of this primer, short nucleotide sequences. These are the steps involved. Here next part we will discuss about the exposure of DNA strand as well as leading strand and lagging strand of DNA. The next part. Thank you.